Welcome to our next seminar in this series in this summer 2025. We speak about plants. We speak about different treatment technologies, phytosensing, some new ideas in this field, because what we learn, this topic become very hot, very, let's say, recently very relevant because of climate change, because of appearance of new technologies in this agriculture field. And we had already some presentation in also related to, for instance, quantum biology, different spin, proton spin, electron spin phenomena that play a great role in the plant physiology. We had presentations about, for instance, classical approaches like inoculation, so it's typical for agricultural science. We also we learn about plant and musics, the Lauren presentation. And today I would like to present our guest, Victor Prevost from company Genodics in France. And as I learn what they do, we invited Victor to give a talk. It's also very, very interestingly, I'm absolutely excited about your talk, Victor. So first of all, hi.
Hi.
Okay, and I think, Victor, so it's a short introduction. I think you can start your presentation, and I have already a large list of questions, and after your presentation, we probably can have a sort of answer questions session and a little discussion on this or other topic.
Okay. Well, okay, so I'm Victor Pelot, and so I'm by formation an engineer in biology, so I work in labs in microbiology and cellular biology at CNRS in France. And so I got interested in the topic that we're going to talk about today with Genodics. I met this company. I heard about what they're doing, like working in agriculture with sound, and that kind of fascinated me because I like music and stuff like this. And I was intrigued, and we started to build this project around maybe a thesis on the topic. I was just finishing my master at the moment. But yeah, so the thesis didn't happen, but then we built a research project together, and that's how at the end of it, I joined the company. So that's a short summary. So, do you want me to start the presentation right now?
Yeah, please start there.
Okay. So I'm just going to share it. Here we go. So yeah, so the company name Genodics already gives some hints about what we do, but it's a name made from genes and ode, like in melody. It's basically the sound of the melody of the living of the genes of the organism, and we're going to see why. I titled this presentation "Protein Harmonics for Plant Protection." It's not only poetic; it's actually what we are dealing with. So as an introduction, we're not going to read all of this. It's just for like broad information, because I feel that the topic of plants and sounds, and even more for music, is not very well understood or very well described in the academic world. It's kind of a niche subject, and especially in France, we have a very hard time to interest academics on this topic. So I need to do this little introduction to review what has already been done as a research on this topic. There is many things that has been done, and not all is really well known and accepted. But still, like it's been demonstrated, like mostly there are a lot of Chinese paper about this. There are a lot of Chinese paper like showing this for many years now. So they are in advance on these very, very questions. They're using sounds in their agriculture for many decades now. And so at different levels, it's been shown that plants are sensitive to sound and vibrations. and they can regulate their activity and show some interesting benefits from being exposed to specific frequencies and frequencies. So, for example, like the first paper I mentioned show that some various physiological process can be modified or stimulated, basically, like germination, flowering, growth, and also defense mechanism. And on the wide range of frequency from 250 hertz to 1 kilohertz, show that there are many things happening at the physiological level. And some specific case, we're going to go back to this later. But also on a molecular level and at a cellular level, there are some changes that have been observed. Like on this second paper, they talk about the reprogramming of genes or of genome and the expression of it with sound. And also still in the pathway of stress resistance. And they identified some specific receptors for how the plant can react to vibrations and also maintain this the cell wall integrity. This is the interaction with the air, so the main receptor of air vibration, airborne vibrations. And here also I mentioned this French paper that was released in 2023 that talk about also this aspect of memory of plants that they have with sound. Maybe not only with sound, but it's been shown in this study. And recently also, this Israeli team, they shown that plants are emitting ultrasounds in the same range of frequency as some insects. And flowers can respond to frequency emitted by insects, pollinator insects. So all this show that there is a relationship between plants and sound. And this it's not obvious when you look at a plant that there is this connection. They look very static, but we have to understand that plants, they are not moving, so they have to, in a way, they have to be very hypersensitive to their environment. They have many channels to perceive the surrounding information. So, and also like the sound vibration shown to influence the production at the root level, and perhaps like changing the way plants communicate within the soil with the network of the microbiome and the mycorrhizum inside the soil. So that's also another fascinating aspect of that. So there is all the reference here if you like to take a look at this. But now we're going to just look at this paper briefly to have some context. So this is a meta study that showed. I like this paper because it showed that we have a lot of a big range of frequency, a big range of exposure duration, and also a big range of effects on the plants. So that shows also the modularity of this approach with sound. It's still like a subject that is still in exploration, but we can act, as is shown in this little drawing, that at the transcriptional level, epigenetic level, and also in the defensive mode for the defense hormones. So it's very like a wide approach, and yeah, so that also shows that there are a lot of data on it, and also a lot of applications that are already in agriculture. And you can see the countries that are most cited in here, that are China, Korea, Iran, and Turkey. So it's not very popular science, I guess, in the Western world. There must be some reason for that. So this is another aspect that we talked about, how the flower respond to pollinator. I think this really shows the specificity of the response and the perception of a signal. It's not that the plant is just perceiving passively a signal from outside, and every signal will do. No, here we have a different type of frequencies, for example, from the 1 kilohertz from the bee and the 35 kilohertz from the bat ultrasound. It's not the flower is not responding the same with the bat. It's not responding at all. So it's also like we have this idea of a specific response to a specific sound. So now we can think of a more like of a signal, the perception of a signal. I think that's very interesting for what we are doing. This is the French paper, so it's all in French, but I will translate as I go. What they've shown is, we talked about it, like the memory of the, so they show that the inoculation of fungi on the leaf of Arabidopsis is slowing down the expansion of the infection on the plant, is slowed down when the plant is exposed to certain frequencies. So that was very interesting. It's the first time that a French scientist team in academic contest did that, and they have a lot of difficulties to put that experiment together, because it's not really well admitted in France, in French lab, that we can do this. But nevertheless, they did it, so that's great. And they show that the plant react positively in terms of immune response to the frequency, and also they show that this immune response, this better immune response, is still present like 3 days after the stimulation with the sound frequency. So they have this memory: when they stimulate once, 3 days later, the plant still act differently than if there was no sound. And that's very interesting. How is it stored, this memory? What's happening? That's a lot of questions. They have some ideas on how it happens. 3 hypotheses mainly, like the presence of trichomes, that's little hair on the surface of leaves, but yeah, that doesn't account for the leaves that don't have hair. The microtubule and the organization of the cytoskeleton of cells, that's also another, because we know that these microtubules, they have some piezoelectric properties, meaning they can produce some electrical activity when they are solicited, like mechanically. So that can be one aspect. And also ionic channel that are more or less open when the sound is emitted. So those, like, some hypotheses. You see here some figures also that model the way the leaves are resonating with the sound. This look like these Chalney models of vibration. It's very interesting also to understand how the plants can perceive sound, because often when you talk to people about plants and sound, they say just, oh yeah, how plants don't have ears, so they can't really understand sound. But now it can be shown that they have receptors for sound, leaves or something else. So yeah, and this is a summary of all the various ways plant communicates. So that's a big summary, but that's what we said before. But you see that plants are very solicited, like from the outside environment, many type of sounds, many type of vibration, and they react to all of this also in the soil, and they adjust also their own production of chemicals. So like the communication between plants with chemicals is well known, but we don't see plants communicating with sound, but it's becoming more and more plausible. Also, like I talk about this paper that showed plants produce ultrasounds. And that's very interesting because they produce ultrasound, like loud ultrasounds. It's not like you have to be very close. If you hear ultrasounds, you can measure this at several meters from the plant, and it's the same frequency with the insect and other stuff that have this kind of range of perception and emission. So that's triggered the question, why and what is it doing? So it's a vast field, but this only goes to say that plant can perceive sound and are likely to be using sound in their daily sessile life. When they are fixed someplace, they have to use sound from the surrounding to modulate their own physiology, metabolism, and activity, and the way they're going to grow and what they do. So this is becoming more and more the new context of thinking about plants, not just staying there and having only chemical interaction. Now, to our subject, that is so genodics, and what is the core of what we do. So basically, we we use sound frequencies to stimulate natural resistance in plants in agriculture. But it's not exactly the same way we've seen in the papers before. Here we are targeting specific metabolic aspect, so specific response from the plant with specific sound sequences. And those sound sequences, they are produced and deduced from biology, from protein sequences. So just a summary to say what is a protein. A protein is like a big molecule produced in every organism that is functional. It's kind of tiny. There are like many proteins in the organism, 20,000 around that in the human body, and they're producing many things. Like the muscles are proteins, hormones can be proteins, many enzymes. They're doing like little robots in the organism, doing all the type of function. And DNA, I think everybody knows what DNA is, but DNA is the guidebook or the recipe book to how to make proteins, basically. When you take a gene, you take some extraction from this gene, is the recipe for this specific protein. So DNA is the book to how to make all those proteins. This being said, so here a protein, we can understand it as a necklace made of different type of pearls of different colors. And in every living organism on Earth, we have only like 20, 22, but very rare occasion, but 20 colors of pearls. And all this arrangement of these pearls, when you make this necklace, make the specificity of this necklace, that is the protein, and give it this function, this shape, and and what it does. And each pearl, that's what they call amino acid. Amino acid is this substructure, sub-element of the necklace protein. So what you see in the table, like on the left, each letter corresponds to an amino acid. That's the way it's written in biology. And so the idea, and we're going to explain how we get to this conclusion. The idea is to convert each amino acid into a corresponding frequency, so a note, based on its mass. And we reproduce this sequence of frequencies in audible range, so we can work with this. And so why are we doing this? What is interesting is that frequencies, like in music, they don't follow each other. The notes in a melody, they don't follow each other randomly. There are some rules, and basically music, although it's very cultural, there are some basic fundamentals that is called harmonics. And harmonics is a physics property. It's like when you have one frequency and you double it, you have the first harmonic. When you triple it, you have the third harmonic. You make it 4 times and you have the fourth harmonic. This is physics. When you produce any frequency in the material reality, then you have these harmonics come in there. And those first harmonics are the one we find in the music, like the octave, the fifth, the fourth. They are very basic, and we can find them in every music in the world, every music system. So music is based on that, but then extrapolate into the culture and different meanings. But what is interesting is that this harmonic relationship between notes, we find this relationship between the frequencies of the amino acids or in our biological proteins. So that's what this graph is showing. When you take many, like 2,500 proteins from humans, and you look at these melodies, and you look at what are those harmonic relationship between the notes, you find that some peaks, like almost like a music. It's like you have some harmonics that are, it's not random, it's not uniform. You have the fifth, the unison, the octave, they're all like overexpressed. So that suggests that there is this harmonic relationship within the living. So yeah, that's a lot to take in, but that's the core concept of it. Now I'm going to talk a little about how did we get to this conclusion? And it all starts with fundamental physics, with the French Nobel Prize, Louis de Broglie, that introduced this hypothesis at first, that to each material particle, like everything that has a mass, an object, must be associated a wave, an actual wave. So we know this very much today. We think about light. It's very known experiment about the duality of light, that is both wave and particle. And so this is a concept we get to understand today, but then it was not as clear, and he also like developed this hypothesis into a formula that you can correlate each mass to each frequency. And so he received the Nobel Prize because experimentally in 1929 it's been shown that we can show the wave property of an electron. Electron then was really used to be considered as a particle, a very, very solid object. It's like saying you can show the wave property of a table. Doesn't make sense. But then they were able to show it at this scale. And so this validates his theory. He received the Nobel Prize. And since then, we've been able to diffract, so to show the wave property of many, many things that we've had were objects, like atoms and then molecules. And I think right now we are having this huge artificial molecule. I don't know how many thousands of atoms is made of. This very big thing that we know is an object. And we are able to show in specific condition the wave nature of this object. So this is like the core of quantum physics, of fundamental physics. Is like this is we see things as objects only for reasons, but fundamentally, when they go back to the very primordial stage and very small scales, and it doesn't have to be object, just a way of framing it. So there was this young researcher at the time, Joel Steinheimer, who is the father of Genodics, who was working in the lab of Louis de Broglie. And so he has this question for his thesis: how to resolve the masses, the repartition of masses of elementary particles. So like subatomic particles, why their masses are what they are. Is there like a rule underneath? Is there an explanation for this repartition? And so he took this approach of Louis de Broglie and converted these masses into frequencies. And he was also a musician, and he became also a professional musician. So he had this idea to look at the harmony in it. And he shown that that the masses of elementary particle, the more they are stable, the particles themselves, the more they have a long half-life, the more they are with harmonic properties, basically. And so this work was presented in Academy of Science of Paris by Louis de Broglie himself, because it was recognized as like that was an interesting way to look at it. It was correct and interesting. And so so for Joel Steinheimer, like the image you see, this is like two particles colliding in an accelerator, particle accelerator, like I think it's a muon and a proton. And so for him, you can see it as an explosion of many tiny particles from two particles colliding. But he would say it's like more like a chord, a musical chord, like two notes making like a musical chord. So it's like a minor A here, and not like this collision. That's poetic, but also it makes sense on a frequency understanding of it. And so so we applied this. Just one example of what we do with this. So maybe I'm going too fast, but Joel Steinheimer, then he applied the same idea to biology, because he recently heard of these two Japanese geneticists that were saying that they were working with DNA, and they had to remember like long sequences of these four letters that are making DNA, A, T, C, G. and they have to remember it. And at some point, they develop a system where they will just whistle notes for each letters, and they will remember it more by whistling than just with the letters. And the idea is that they were like patterns, regular patterns in DNA that you can hear when you whistle it. And this give Joel this idea to apply this to proteins that we described before and to amino acid, and to apply this formula, this correspondence between mass and frequencies, to proteins, and to see if there are this harmonic correlation between those elements. And so yeah, he found out that there is this those correlation. He started working on that, and he developed this method, and also produced a patent to show that this is a way to we can use these frequencies in a way, and that's what he shown, that basically at the theory that came after all this, showing that there's this vibrational aspect also in the living organism, that during the synthesis of proteins, we have a production of let's say a frequency or many frequencies, and these frequencies, these waves that are produced during the synthesis of proteins, they produce new constraints, new vibratory constraints on the synthesis of this protein. So the idea was then to reproduce this signal, this sequence of vibration, to kind of constrain biological system to produce protein in one specific way, basically. So it was to reproduce this already happening in the cell, the thing that was already happening, and that's how it came to this idea of the patent. That is a way to convert protein into frequencies that we can broadcast to organism to orient their a specific aspect of the metabolism. So it's a lot of information, but what is interesting is just this tiny example. I think everybody know La Primavera de Spring from Vivaldi and how the music goes. And I go da ta ra ta 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 ra ta 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 ra. Yeah, okay. And so what you heard is not a remake of this famous song, but is the transformation of a protein, the actin, into its system of frequency. So that's very interesting, because actin is a protein that is present, is everywhere in also animals, but also in plants, and is very, very much produced in the spring because of the growth of the tissues. It's structural protein. And so there is this idea that maybe like why is this correlation between this sound, this melody, and Vivaldi composing La Primavera? That's an interesting idea. Maybe he was influenced by this, we don't know, but just to show that when we convert proteins, that there was what you heard is a protein that is converted into sound, it makes sense musically. So that was like maybe the most important thing that triggered the curiosity on the topic, is like it's not random noise, it's really interesting. There are many examples like that. So that's the concept, that's where it's come from. And so to produce this patent, it took a long time, like around 10 years, because you need validation of the concept, and to be sure that was something observable and repeatable, that something was happening. So it went through this big process of validation, and many studies were done. For example, this one on the natural luminescence of some bacteria called Vibrio fischeri. And so it has been exposed to the melody or the sound of two genes, two proteins, the lux A and lux B, that are involved in the luminescent aspect of this bacteria. And so we see the result that depending on the time, the stimulation time, we have a difference in luminosity. So there's a correlation between emitting this sequence and the actual measured light produced by this bacteria. And so there are many aspects. There is like the duration. There is like the in time, also in the duration of exposition. So you see, it's not always because it's more exposition that it's better, and there are some peak of expression, and so it's not like a linear process also. But that's one aspect on bacteria. This has been done also in a INSERM lab in France on the interleukin, is a protein that was trying to be down-regulated on a human cell, cultivated in lab. And it's been shown that the amount of this protein is reduced when understimulated with the same approach. So this was all published within the patent. So we have like bacteria, we have human cells. Here is yeast, so fungi, yeast in bread, and so that when stimulated on some, I think the proteins were like of the respiratory cycle of the yeast, and show that there are more volume of bread because the growth of the yeast is better, and less acidity within the bread. So this is all like preliminary studies. This is the work through which I joined the company, that we did in the lab in the area of Paris at the University of Cergy-Pontoise. And so we work on this model on growing some peas, and to try to stimulate or to help them in condition of hydric stress. So to stimulate one specific protein that is called dehydrin, that make plants more resistant to hydric stress. So on the graph, you have like two sets of population, one is darker than the other, and two curves that showing the tendency, the trend of each set of individuals. And so on the axis, the horizontal axis, you have the weight of each pea when they grow, and you see the weight is bigger, so they grow bigger when they are stimulated, and they grow smaller when they are inhibited. So that's been replacing a lot of time, and has been published in a review in 2020 and also in 2021. And that was like, we measured the macroscopic aspect, how the plant behave itself under this condition, because we produced stress for the plant and then broadcast this sequence, this musical sequence that is targeting the specific dehydrin. And then we are able to measure the amount of this specific protein within the tissue of the plant. And this is what you see on the right, is like the spot. They are like the one here on the where there is the arrow on the top, is the protein that we have targeted, and is like 30% more present in the sample that has been stimulated. So we can see also that at the molecular level there is more of this protein in the sample. So now, from the company activity is mostly in agriculture for like 15 years now, because it was easier to work on plants, and also we had to make the practicality of this approach and the potential to make it like the proof of concept on the field and in the real with real actors and in production context. So wide range of activities, wide range of application. You can see like in greenhouse, you can see for tomatoes, in playing field, also under the water for oysters. That we don't do that anymore, but we did it. It was interesting, like viruses on oysters. And so we have many results. I'm not going to go in every detail, but on apples, because what is interesting here is that to show that we have good a good response on many type of pathogens, like on fungi, like the tavelure, like on Sharka is a virus. Mosaic also is I think is a virus. And so we have different organisms are replying correctly from that. We can see like from the courgette is a zucchini, we have this graph with the colors. Is the more we get away from the device that is broadcasting the sound, and the more like the mortality increase, and the quality of the fruit is lesser. So that's interesting. Also, like on endives, we show that the more we expose the plants to the appropriate frequency, and the better is the quality, the less is second choice quality appearing, caused by this Erwinia fungi that appears during this growth of endives. Here's just some testimony of producers. So for example, this person, Edwige Lovage, is so growing vegetable, and there is like this need for many people now in agriculture to reduce what they put in the field on the plants. So it's also a matter of economics, but mostly on the environmental concern. So here she's very happy because she's not using any chemicals, and she has like beautiful fruits, beautiful vegetables. So I mean, that's not the only thing she does that using our system, but it's a whole way of cultivating the plants. And the idea is that we are here to help specifically on disease and on like stress, environmental stress for the culture. So because when you have this idea of a system without chemicals, you also adapt the way you process the whole thing. But sometimes you need help on specific topics, specific problematics, and that's where we come. So this is a wine producer, Philippe Suquetas, in the southeast of France, and he had here is the area is very very dry, and he has some disease also. The fan leaf virus, I think in English is like this. But yeah, since we installed the device, that you can see on the picture is this box with a solar panel and just like speakers that are emitting on the field. Since he has this, he really reduced the disease. And also we work, it's not written here, but we work on the stress caused by the lack of water. And this also show a real betterment in the plant growth and the wine and the grape production. So what is very interesting that the strains that are affected, they remain affected, but it's slowing down the disease, and it kind of make it like because the pathogens also we see it like this has its place in this ecosystem. We don't try to destroy a part of the ecosystem. We really try to manage it and to reestablish this kind of communication between different aspects of this ecosystem, that the plant that is cultivated is part of the ecosystem, the fungi and all the viruses and everything are part of it, and also the human that is cultivating is also part of the ecosystem. Everything is part of the ecosystem. So that's the idea, that we try to make this harmony, as we talked before, between all aspects of the system. That's the fundamental aspect of this approach. This is a strawberry producer in Bretagne, and so you can see he has very good results because his plants are stronger and with less disease, and he has like a very good betterment in yield. So yeah, it's interesting that first we started with the device to broadcast the sound. It was outside the greenhouse, and he was like, "Yeah, it's better, but it's not quite this and that." And we realized that the thickness of the glass was preventing a huge part of the sound of the information to reach within the greenhouse. So then we put the box inside, and he's going to install a second one in the other greenhouse that he wanted to cover with as a wall. So that's very interesting that we're still learning how the information goes from here to there. And but here is really striking results. It's very happy about it, and we work on the powdery mildew on strawberry in this case, and he's very satisfied. This is also like vegetable growing. So same, the reduction of the use of chemical treatment was the main focus on the endives and this fungi sclerotinia. And so we managed to reduce drastically the use of chemicals and even to avoid them for like 2 seasons, and to still have a good production. So yeah. Wine again. So we work mostly in wine, in grapevine wine production for historical reasons, because in 1998, I think the one chemical was banned, that is the arsenic was banned it was used to prevent this disease called ESCA, that is a fungi that is eating the dead wood in the plant, in the wine, in the vine. And so this was stopped, and then wine producer, they were like without any solution, and the plants they keep dying. And so we started with this. I mean, I wasn't there at the moment, but the company started with this application for this ESCA disease and had a very good result. We're going to see the result just after. And so this is why it started like this. This is what started the company and allow it to develop other applications. So this is one of the first client that is, I don't know for how long he's been with us, but like despite a very hard a very humid season in 2024 in organic farming, nonetheless, they didn't have mildew on the grape. So it was very a unique case this year. So yeah, it's not only because what we do with them is like there are many things they're doing and they're doing right, but yeah, we help in that direction also. Another wine grower, like in most of it is in France, of course, and he was working against it's like this disease that is transmitted by a worm that is getting inside the plants, and so we are able also to work on that. I mean, what is interesting is example that he also speak by himself about how he understand all the process and trying to explain, and because there is this aspect also that is a different type of agriculture, and so a lot of people love to talk about it, and it's interesting. It's a curiosity even for people coming to see the vineyard, and they're very interested into this. It's a very different approach, very surprising one, and also like the quality of the wine is also at a different level with this approach. So that's also one aspect, like the human and social and cultural aspect of it. And yeah, so this is also like on wine and in the south in Camargue. So again, organic farming with the same good result with the same device. So this is just some results that we had on mildew. You can see like on the top of the drawing, this is where the box that broadcasts the sound was situated, and this is a 10-hectare field. And so you can see that there is no impact of mildew, or very few, near to the device, and at 150 meters they start to reappear. So here's the same curve that you see on the right, showing that the more you go away from the device, and the more you have a mildew. So we can show that there is like a gradient of information. The information is like spreading further and further, but at some point it stopped being significant for the plant. And we understand it as we do it by hear. Basically, we see that if we can hear it, we know that the information that is within the sound that we display, this information is reaching the plants here. And then we don't know, because plants don't perceive the sound as we do. But here we have a good idea that there is a limit to the information. This also has been published in this review, Responsibility Environment, that is from École des Mines. Okay, this is the result that we have the most on the SK disease, to show that all cumulated many years, different. It's all around France. It's been for like this pulling all the data together. We have, on average, and a significant average, a reduction around 60 or 70% of mortality each year for in the vineyard. So this is that we repeat year after year. It's been like 15 years now. So we are very, yeah. And this is also interesting because we have a lot of data on that, because I said that there is no alternative as a treatment. So it's quite easy to compare with and without. And that's one difficulty when we work on different subjects in agriculture, that we have to be sure there is a production. We don't want to compromise the production of the agricultural system. And often there are already some treatments in place. So it's not like in a lab that you can do your experiment and compare this and that. So here we had a clear comparison. Also on the right is the graph that comparing two fields, two vineyards, but with identical typology, in the same exposure, the same moment of plantation. But then you can see that one is always more impacted by the disease than the other, and the one with the music is the one that is less impacted. So, and that's constant, despite the variation that is every year. We know that fungi have like many year cycles, especially this one, SK, is like a 4 or 5 year cycles. But despite that, and despite the cycles, the difference remains, and we have better results with SK, with the music. Okay, I will finish on this. This is among the difficulty of measuring stuff and to evaluate, because in agriculture you need like to wait basically one season to see if what you do is correct or not. This has been done with a sensitive crystallization. That is a method that is used to assess the quality of products. And this is very known wine that is known Domaine de la Romanée Conti. It's very famous and very, very qualitative. And so for conditions were set up to produce this wine. So so I didn't say that, but proteody is the name of this sequence we use. Like proteody is the melodies of proteins. So it was using proteodies, so the sound display, with or without normal treatment, that is sulfur and copper. So we have our cross condition with the two, without the two, with one or with the other. And so this was done. It has been done like in a blind study. Margaret Chappell, who did this analysis with blind, just look at how the juice of the grape or the wine is crystallizing, and can see that from this structure can tell if it's good or there's balance and everything. And so the one that is the most interesting is the third one, that she says that is a very nice product without any flaws. There is perfect product. I mean, we already have a very, very good product because, I said, it's a very famous wine. It's very, very qualitative. But this is like, because when you introduce some chemicals, then you have some the plant needs some time to readjust and to adapt itself, of course, like we also, when we take medication, we need time to rebalance. And when the plant is affected by a disease, also needs time to find its strength again. And so, yeah, here was the condition only with the sound sequences, but no chemical treatment. And it was the best one that has been sorted blindly, so only based on the crystals. So that's also like a qualitative approach to show that we have impact also on the quality of the product afterwards. So that's it for me. Thank you very much. Here are my contacts.
Okay, thank you, Victor. This was an extremely nice presentation. I think I also learned a lot of new information.
Yeah, I hope it was not too dense. I don't know.
No, it's absolutely fine. I have, like I said, a lot of questions. I divided to 3 blocks. Okay, let's start with the first block of questions, probably more on practical side, because I see you have, let's say, a lot of practical work with different agro enterprises. First point, efficiency. So I saw in some graph efficiency is between 5% to 10%. So what does it mean? You, let's say, without applying your approach, so it's a background message, the measurement, and if you apply your, let's say, sound treatment, improvement, yield, for instance, improvement is something like 10% maximum. Is this correct?
So, well, I mean, is there a graph specifically you are referring to? Maybe I can show the graph.
It's in different slides. For instance, there was around East and success after East. In the East, there was about 5% difference. Okay. But generally, I mean...
yeah, well, okay, in general, it's I think 10% is not that... I mean, we have better than this in general. It's with a pill like this, but like, for example, like in the case of the SK on the wine, we are around like 60-70%.
But wait. SK is a disease, okay? Disease is a little bit, let's say, I will ask this question. But now, for instance, if you are going to improve biomass growth or you know, fruit building, so
we are mostly like this is I did. Maybe I should check like from the bread. Maybe it's different, but it's around mostly 30%, more than 10%.
30%.
It's
3-0, yeah.
Yeah, like the difference in the amount of protein was 30%, and the difference in the growth, I think, was maybe a little less than. Yeah, I have to check this on the specific graph. But yeah, I mean, that's wrong with that. Maybe not in biomass, but you can see that with the strawberry, the production of fruits is from 2.7 to 3.4. It's like
yeah, I think there was strawberry. There was 5.6 without, and with your approach 6.6, something like this. So it sounds like 10%.
Wait, I don't have those data in mind. Sorry.
Okay. Because so 30% is a huge. I mean, it's really huge.
Maybe not on the total biomass, but yeah, 2.7 to 3.4. So how much is that?
But okay, let's say something like up to 30% at least what people can expect.
I mean, what I want to say is that it depends. It depends on the application.
Yeah,
It pretty much depends. I wouldn't say it's limited to this or that. Yeah. So, for example, like for me, what is most significant is because always we are focusing on what we want to obtain, basically.
Yeah.
And so, in the case of protein stimulation, it was like around 30% in this case. That was measured like this. I think it was around that also, like in the mildew. But from the standpoint of the company, for example, when we develop an application for like a disease, I know it's not exactly what you're asking, but that's what we have mostly done. So it's what we know better. When they started, they had this contractual thing that if the result is less than 30%
okay.
of what is expected, then you get reimbursed entirely.
Yeah.
So because we observe that we have more than that. I mean, in the worst case, I mean, there are worst case when it doesn't work for many reasons, because it's not the only factor in the environment, of course. But in the worst case is around 30% or a little more. But at the better is like 60%, 70% in the case of the disease, for example.
Yeah, this is let's say this pest stress diseases is absolutely hot topic now. For instance, ESCA is okay, it's impressive. What's about Peronospora? For instance, in vineyards, Peronospora is I think is a first or at least second.
Peronospora. What is the name of the disease? You know, because it's maybe Peronospora. What is? Because we work with the French words, so I'm not sure it's the way it's.
I have to check probably. ESCA is another name for Peronospora. I know it is from our
maybe it's just a mildew, no? Maybe mildew.
Yeah. But okay, I will check it properly.
For what I see, it's mildew. I mean, obviously for us in the wine, the biggest problem is mildew. So
yeah. Is normally is a Cooper treatment for it.
Yeah, exactly.
Yeah, okay.
That's why we talk like
yeah.
Well, and copper is used for many things, but sulfur and copper. And so we have good results, but it's not a simple disease. It's very complex, and I think it's not only one fungi implied in it. It's like every fungi disease is very. So I show some result on that on one mildew with the graph with the gradient of effect. So this was on mildew. Also, we have limitation, and we saw it in the previous years. They are very, very humid, a lot of rain, and so wine was very, very impacted by mildew. And so we saw that right now, with what we're doing, the sequence we are using, it's not we can't use just this to prevent this disease. We have to accompany it where it works the best. It's not the only treatment applied. We have to. When you apply this with other stuff, it make it better, and it make it like maybe they couldn't pass it, but they did because many technologies have been used. But for now, there are not a lot of cases when we can use only this, I think.
What's about, let's say, greenhouses, controlled environment agriculture, vertical farms that are specific, for instance, fungal infections? Did you test for, let's say, because it's dependent species, but did you test generally, let's say, some antifungal treatment in this CA?
We do like oidium, mildew, botrytis, all this disease. We work on that on a regular basis. So that's what we do in greenhouses.
Mm-hmm.
And it depends on plants. Also, what we realized, because one of the most significant one, we worked in Martinique in a greenhouse for tomato, and so, but the thing was not really. I mean, it was yes and no. We didn't know really if it was completely okay, and it turns out that the bacteria strain, was not identified properly. So that also has a huge impact. So we are, of course, limited by the knowledge we can get on on what is the infection about. But once we get the right information, it's we work on all type of application, all type of.
Okay. Then let's go to, let's say, next block of questions. Technology. I mean, I saw your emitter device look like a box with 4 loudspeaker or emitters. It's look like it's low power because it's powered only by a solar cell. And it seems, at least in one of the first slides, you say it is a low frequency. To generate low frequency, you need a lot of energy. It's probably something like piezo in the kilohertz area, or 10, 20, 40 kilohertz area, probably.
Yeah, no, we emit in audible frequency. We don't focus on low frequency. I don't know. Yeah, we all bring it. I mean, the calculated frequencies that are like the theoretical frequency that are in the organism, they are very, very high frequencies because very tiny particles. So they are like 10 to the power of 25 hertz. It's unthinkable frequencies. And so, but we keep this information by transposing all this signal. And so we use like around 400 frequencies, like between 200 and 600 or 800 hertz, like range of frequency for the human ear. And so we don't need that much power. There is a battery inside that is charging continually, and that is allowing us to send the sound around like 80 decibels.
So quite loud.
It's pretty loud, and it covers like let's say 15 hectares, like a circle of 200 meters in a region.
Does it distort, let's say, people if because they have to work in the sound reason?
As I said, it's part of the environment. For us, it's part of the environment. So we have to be aware of that. If there are some people living nearby, and that happens, it's not always because like we work in a vineyard is usually remote and it's like nothing is around. So we try to put it at the middle of the yard and not to not to disturb the neighbors. But sometimes it happens, so we have to modulate to put it lower or to move the device. That's a big part of it. But some people like it and they go outside specifically to listen to it when the sequence is broadcast. Also, it's not always broadcasting the sound. It's only like 2 or 3 times, something like 10 minutes, 2 or 3 times a day.
So short.
It's the thing is we transmitting information. We're not transforming. It's not the mechanical energy that is modulating in our view. It's really the information that we
that's exactly my next question. Yes, if this really short term, but okay, let's keep this device first. It seems you emit for 3 minutes, very loud, also 90 decibels.
10 minutes, 3 or 4 times, 2 or 3 times.
Yeah, 3 or 4 times. Yeah. Is this a signal or harmonic frequencies or a sort of music what you're emitting?
It's like when I shown here in the presentation, the melody like this. That's what protein melody looks like. It just notes look like a melody.
It's a melody. It's not a single sound.
Yeah. No, it really resembles a composed melody. That's what is kind of intriguing. It sounds like music. It's so weird.
Okay.
That's because also when you play it, because you have the notes, but you don't have the rhythm, the structure. But when you play it, you recognize there are patterns of notes. It's like it makes sense in a way. So that's very, very interesting, almost disturbing, but it's fascinating. It really like you hear there is a coherence. It's like those guys whistling the DNA. It makes sense for them. They can remember because there are patterns. So it's not what I'm saying is not random when you look at this like that.
Okay. I see. I know. I mean, for instance, this ultrasonic signaling system, in plants. Read it carefully. We know people in the laboratory, academic research, they work on this. But this look like really a fixed frequency sound. So it's things like this. And if you say, okay, the harmonic signaling, the melody, it's really interesting. Okay, let's say how much your work and research was inspired by, this topic. Plants like musics, they listen musics. How much is inspired by this, let's say, popular topic?
Well, I can't speak for Joël Steinheimer who started this approach, but yeah, I don't think that was his focus at all, because first he worked on human proteins, so he really was fascinated by the fact that there are this musical regularity as a musician, as a professional musician, that there are this musical thing. The first molecule he worked on was the one block of hemoglobin,
Mm-hmm.
and he was like, okay, this is musical, doesn't make any sense, why is it musical? And in some like weird context, some friend of his he was in contact has needed a blood transfusion for some reason, but she couldn't have it. It was at the time where there was this contaminated blood in France with the HIV virus, so it was they couldn't have. And then she listened to this sequence, and she felt better, and she had improved results. So that's how it started, basically.
Okay.
The idea to work on plants is really to take some step back and to kind of get more objective,
Mhm.
basically.
Okay.
Yeah, it's not we trying to, I mean, it just happened to be like this because we thought it was useful for quality of food, of production, and to help agricultural domain that need some solution. And also because if you try to work on something else than plants, then you encounter some difficulties,
Yeah.
medical and everything of this. So it's mostly coming from this. I don't think Joel had this in mind to, but I think I know that some sci-fi novels were inspired by his work also. That's why maybe.
Okay. Next, go to more theoretical points. For instance, yeast. I know that, for instance, if you fermentation process, for instance, very much sensitive even to vibration. And if you, for instance, this patent validation slide, you said, well, that you validated. Question is following. Okay, you have signal, you have sound, you have vibration, and you have melodies. Question is, I can hear myself now. Okay, question is, what let's say, which specific piece of this let's say. Signaling, sound or melody impact different organisms. Also in yeast, I can assume it's properly vibration.
Yeah, that's a very very fair question, and I mean it's a hard question also because we don't have all the answers. But for example, what have been done on the bacteria that make this luminescent bacteria, it has to be done without the membrane, I think, of the speaker. So it was only the electromagnetic information.
Okay. This is something different. Yes. This was my.
Yeah. So it was So I mean, we didn't have the means to investigate that for now. It's also was something we have to investigate, how information goes from this to that. But apparently, it's not about the sound. The sound is just a carrier, but it's about the information itself.
But look, if you have, for instance, in the field 200 meter, or let's say in the middle something like 100 meter, electromagnetic waves with, let's say, energy what you produce by battery, I don't think they.
No. I think it's just it doesn't matter. I don't think it matters that much what is the carrier of the information. I think just
we should think about this.
Sound waves in the air can be perceived by the plant at a distance, and they then interpret it as they perceive sounds. They can perceive it, and they integrate this information. If you bring them closer and you can expose them to the electromagnetic wave, the information within the electromagnetic wave, then perhaps they can also interact with this. I don't know. I mean, we know that in experiment with speakers, you have to put the plants far from the speaker so they don't receive the electromagnetic thing, so do not bias the study. But for bacteria, it seemed to be working with the electromagnetic field.
But bacteria, yes, I mean, there is
So
sensitivity
plants, I
to
don't know.
electromagnetic fields now, yeah. But
We haven't done this study with the plants.
plants, they are sensitive, extremely sensitive to vibrations. And my assumption is, well, that you create a sort of vibration.
But the thing is, like, for example, in the experiment we did in Paris in the lab that we published, there were sounds in both cases. It was stimulation and inhibition, because we also, like, with the sequences, we can like invert the information basically, because you have a musical pattern or melody, you can invert it to make the exact opposite from a center point, and then you have something that is not in tune with this specific melody. Well, this is stimulation, inhibition, and so both has this vibration, but the information was different, and we see the most difference in this case.
Well, can you please explain, if you say information, what sort of information you have in mind? Because, if it's melodic, for instance, Vivaldi, what sort of information is provided by Vivaldi music?
For us, I mean, there are two levels. I'm going to talk about two levels of information. One is in the melody itself, so in the intervals between notes, the structural information, and also the frequencies themselves. They are also information, these specific pitches. What the theory that we use and that we established, also he said that information can travel through various scales, because the pattern, the intervals, they don't change. If you play some melody low in the piano or high, you can recognize the same melody, even if it's not the same notes. And that's especially true with the same, what is that? I forgot what it's called in English. But the tempered scale in the music, the tempered scale, the 12th tempered scale. So that's because all the intervals are the same, so you can play that, and it makes sense to displace a melody. But even without it, you can take a melody and change it different pitches, and you can still recognize it. So this is one information, and then where it is in the scale is another information. And for us, the most significant is the information that is traveling through the scales. But for an organism to be able to receive this information, of course, as for you to be audible, it has to be at a certain place in the scale that you can receive. So that's more a constraint between emitter and receiver. But it's not about, the information doesn't change. The information is the intervals.
Okay. Question is, how much analytically you can work, for instance, if you know some specific diseases or some specific, let's say, plant species, how much analytically you can calculate specific pattern or use, for instance, this or this melody? Or you just have, some experience of using different...
Well, what we do, that we look at the problematic and what is involved, and how spontaneously we know that how this plant is defending against pathogens, for example, or how this pathogen is entering the plant. So we look at this in a metabolic aspect, in a molecular aspect, what they do, what are the protein involved in that. So we try to understand how it works on the protein scale. What are the enzymes involved in the destruction of the cell wall of the plant for the fungi to get into it, or this kind of stuff. So more basic stuff, what is what is involved in the multiplication of this specific bacteria strain of this and that, and the plant, what is its counteraction to that. Then we look into biological database to find these proteins, and because we have this, like I didn't say that, but you can see on the table that we have 20 amino acids, but only 10 notes, because some masses are converging into one note, one frequency. So we have a different grid to read the biological protein database. So we can look at it, and some similarities will appear between proteins from the frequency perspective that you don't see from the biochemical perspective. And so this also gives us a hint on what is, according to what is going on. If you do this, is this stimulated? Is this going this direction? What is, so you have this kind of communication within the metabolism, within the proteome. And also, all this helps us develop this what we have candidates protein, and then we select the one who looks the best in that, and then also we listen to it. But some make a lot of sense musically, and just like, okay, this is maybe a hint that that makes sense. You listen to it, and it's like, yeah, interesting. And we had this big part of our work now, of my work also, to try to evaluate the musical qualities of a segment, what it's first, what it means, and then how to evaluate it. Because for us, like, if it's really makes sense kind of musically or at this level of perception, it had a good chance to be efficient in what we do. So yeah, because it's the law of harmonics, and as said, I see it as a set of constraints of harmonic or vibratory constraints that you put on the organism. So you amplify these constraints, and so you amplify what is underneath these constraints.
If I saw your slide with these crystals to evaluate results, I just immediately remember phytosensing, because the whole idea of phytosensing to receive feedback so very fast. Typically, yes, for instance, in our vertical farm, sometimes if you're measuring dry biomass, okay, we have, of course, to wait the whole cycle, to cut out everything, and to measure all this, this hassle. Of course, it takes time. But idea for phytosensing, you just, for instance, select some specific physiological parameters like subflow, biomass growth, or water content, and you apply your stimuli, and you see in real time or close to real time what's happening with plants. So it's just, idea with probably may make sense to apply it not only biomass sensor but different sensors.
Yeah, but that's also what I was very excited about the biomass sensor.
Look, let's say more to subflow, water, whatever in STEM, this hydrodynamic system, because it's provide a lot of information. For instance, what we are publishing now, this study with one company, stimulation by electric field. And it seems it's a change of physiology just to consume water. So sometimes plant don't need, but sometimes you can specifically change it to water consumption. And we simply verify it by, we see this in STEM, plant switch instead of, let's say, photosynthetic activity start, to consume water, to save water in tissues. And this was immediately visible. So that's okay.
But just a question about that. Because our model right now is on these peas, these very little sprouts of peas. Do you think can you take a bigger plant and that you use for many experiments? Or
sure. Tomato, for instance, we tested with tomato, pepper, cucumber, all these classical plants. And you see this, for instance, response biomass. Let's say biomass, you need something like 1 day probably to see at least some small response and inclination of this biomass grow. But with for instance, stem, so water, stem, subflow, you see one circadian cycle, something like 24 hours. You already can receive a lot of useful information. You repeat 1 week in different conditions, and so you have your statistics. So it's but it's, as I saw this picture with this petri dishes, it's just remember, okay, phytosensing. Last block of questions, Victor. Company, academic life and company. How difficult is for you to switch from lab, let's say, to a company?
Well, right now I'm full-time with the company, but we had recently a project with the lab. So for 2 years I was 80% with the lab, so it was a different I don't know, it's just because I think as a starter, when you try to do the thesis on the topic, I thought it was going to be easy, because there's a lot of labs that are working in agronomy in France, agriculture, in INRAE and stuff like this. They are huge structures. So I thought naively that they would be interested to this approach, also because it wouldn't cost them anything, because the thesis is funded by the company and the money from the government, stuff like this. They have this help that we can do. So, and then rapidly felt like many doors were closing just in front of me on this specific topic, surprisingly. So that kind of sorted it out, and so I went back to this laboratory in Cergy-Pontoise near Paris. So it was a lab when I did my biology license. So there was this teacher of Olivier Gallet. He was the director of the lab at this moment, and yeah, I asked him because I don't know, I knew him as a teacher, and he was a very open-minded, interesting character. And he said, like, okay, go search for a lab because we are not specialized in biology, in vegetal biology. We do matrices, we do cell biology, microbiology, but this vegetal we don't really do it here. So your topic is maybe better somewhere else. But if you don't find anything, come back to me and we talk. So that's what I did, basically. I came back to him and we talked. And so yeah, I think and then it started like this because he was one person that would take the risk. I mean, I don't know what the risk is actually, but he took it to start this experiment to just check if there is something to do with this topic or not. That was my approach. I mean, as a scientist, I was like, okay, is there something in this topic? Because I met the company with Pedro Ferrandis. We went to the field together to see the clients of the company and that it was real, that something happening. People are paying each year, so they are satisfied because if someone that is working in agriculture is paying you, it means something is worth something. Because they won't pay, it doesn't make any sense for them. So that was I see there was something real in the field, but then I wanted to comfort myself that this topic was a real topic. And so yeah, so we started like this. So and from this, yeah, we have some difficult moments, but not with this lab. So that's my interaction with the academic. It was mostly through this laboratory and Professor Olivier Gallet. So it was good. It was not feeling weird because we were on the same the same line of thought. But after the publication of the article, we went to Montpellier to present the paper, to present the research. And I didn't do the presentation, Olivier did. And although he's a professor and the director of the lab and everything, some young searcher in the amphitheater were like laughing or like making fun of the topic because it was not serious. And it was very like heartbreaking at some level. It was very uncomfortable and but I mean he took the hit. But that was not good. So yeah, I think I'm mostly spared from this and my relation in the work. Then I still have tried to contact people from INRAE and stuff like this. Some people like the team that did the paper, actually we talked together and we tried to find a way to promote this kind of approaches and it's a question because science is about asking questions. We don't know the answer. If we know the answer, then it's not science.
Oh, science has a lot of taboos. Yeah,
but I think that's not science. That's science.
we learned it in very, very hard way. Okay, they have taboos.
Yeah. But so so that's we're still trying to get with more academic partnership. We do have some more and more, but it's always people. It's not the academy. It's people that are really taking their job as a scientist seriously and trying to really search for new things and ask questions that.
But you're still publishing. It's important. Keep still publishing. Yeah.
I mean, I should I'm late on my publishing, but yeah, I'm supposed to.
Okay, Victor. Then thank you. That was absolutely inspiring.
Yeah,
I will also provide to our partners because I mean it seems to be, I mean it's a very simple approach. You just you emit a sound. It's not 3 times 10 minutes per day. It's not a huge effort. It's low energy, and it seems if you see something like between 10 and 30%, I mean improvement, and especially against some diseases, I mean heart diseases, it's nice. I mean it's absolutely inspiring talk. Thank you.
Well, thank you very much for the opportunity.
Yes. Then, Victor, anyway, we keep our contacts, collaboration. Then, see you then next time in probably next seminar.
Okay, thank you very much, guys.